Hello, children. In this session, we are going to talk about various modes of reproduction in case of bacteria. Bacteria reproduces asexually as well as sexually. Uh, Asexual reproduction is also categorized or known as vegetative reproduction. There is a very slight difference between asexual and vegetative reproduction. So, in case of asexual reproduction, you all are aware there is no gamete formation and only one individual parent is involved. That is, only one individual involved or one parent is involved, and there are no gamete formation in this. So, the first method of asexual reproduction is binary fission. As the name tells, binary means two. So, this bacterial cell, it divides from one and it forms two daughter cells. This is the most common type of asexual reproduction which occurs in bacteria. And as you can see that if this is the structure of a bacterial cell, then there is a bacterial see, you all know it is a prokaryotic cell, so there are no membrane bound organelles are present, and this is the plasma membrane. And this circular structure, this is showing the bacterial genetic material, and that is DNA. This is attached with the infoldings of plasma, and that we have discussed before also, that is called as mesosome. So when this DNA, it replicates. So what happens over here is that DNA, this replication, when it takes place, so it forms the other copy of this. So there are two circular DNA which are formed, and they remain attached with this mesosome. So even the newly formed cell, that also remains attached with this mesosome, with the plasma membrane. Now, what happens after this? That the next step would be the entire cell would divide, and there will be no cell wall formation in this, and the cytoplasm divides to form two daughter cells in case of bacteria, and each daughter cell contains genetic material. So, here from one cell, two daughter cells are formed, and that is called as binary fission. When all the conditions are optimum. So, what happens there in case of E. coli, that in every 20 minutes there are two daughter cells which are formed. That means when all the conditions are accordingly, which are favorable for the reproduction, so this E. coli would form two daughter cells. Actually, this replication of DNA, that gets completed in 18 minutes, and in the remaining 2 minutes this cytokinesis and the cell wall formation takes place, which helps in splitting the two daughter cells. So first thing is that the DNA replicates, and this process in case of E. coli, it takes place in 18 minutes, and after that the cytokinesis and cell wall formation, which helps in separating these two daughter cells, would take 2 minutes further. Now the next step is endospore condition. So this basically is not a method of reproduction, and I'll tell you why. See, from this condition is to withstand adverse condition, so it is not the method of reproduction at all. So whenever there is unfavorable condition, so this endospore, it helps bacteria to survive, and during this adverse condition there is a thick covering of the capsule that takes place around the bacteria, and that helps to protect it. So after reaching the favorable condition, what happens is that endospore cover, it breaks, bacteria comes out, and then it reproduces by binary fission. So we can say that this binary fission divides one bacteria into two. But here if we see that from endosperm, it gave rise to one bacteria, and that one bacteria actually is not divisible unless it enters into the binary fission condition. So this is simply the method of protecting the this bacterium. So during very very unfavorable condition, this E. coli, what will happen? It will die, and the endosperm, it survives, and when it reaches the favorable conditions, after reaching favorable conditions, it will further break and form two daughter cells. The third method is budding. So we have seen the first method of asexual reproduction, that is binary fission, which is the very very common, the most common method of reproduction. Then the second was endospore condition, which was not actually the reproduction. Why? Because bacterium would give rise to one endospore, and endospore will break and bacterium remains one. And in this bacterium where this shell, this capsule is formed, you can see all the cytoplasm, it reaches to one corner, and that remains surrounded by a thick layer, very very thick layer of capsule. So which contains in it the cytoplasm and the circular DNA, which is present inside the cytoplasm. So this endosperm, when breaks, give rise to bacteria. So after the entire process, then again the bacteria remains one, and when it reaches the favorable condition, so then in that case it reproduces through binary fission and forms two bacterium cells. The third, which is very very rare, way of reproduction, that is budding. So you have seen, we have already discussed about budding in the previous lesson. So now this is these are three different methods by which bacteria can reproduce. Now we move further on the sexual reproduction. This sexual reproduction in case of bacteria, it is very very elementary. See, in case of sexual reproduction, what happens is that the exchange of genetic material takes place. So that is why it is very very basic or elementary, because only there is an exchange of genetic material which is taking place, and there are three methods by which this can take place. So we can also say it is a method of recombination of genetic material. So the first method is transformation, second is transduction, and the third is conjugation. In your course, you don't have to see too much in detail about it, just a little bit of introduction is needed. Now the first one, that is transformation, and this is given by Griffith. So this basically, it is a method to prove that DNA is a genetic material. You will read about this transformation in detail in class 12, where in genetics we need to know that what is the genetic part in the body, whether it is DNA, RNA, whatever it is. So what happens in transformation is that the genetic material of one bacteria, it is transformed to the genetic material of other bacteria. So how this all is taking place, that exactly is not known, but this method is used to prove that DNA is the genetic material which is present in the body. The next method you see, that is transduction, and it is given by Zinder and Lederberg. So what happens in this is that there is a transfer of bacteria's genetic material to other this bacteria through a carrier that is called as bacteriophage. So it is the transfer of genetic material from of one bacterium into the another via bacteriophage. So here bacteriophage is actually it's like a, you can say, carrier. So this is nothing but just a virus which infects only bacteria. So bacteriophage is a virus which infects only bacteria. So this transfer is taking place through bacteriophage, whereas in the transformation there was a direct transmission from one bacteria to other. The third type is conjugation, and it is also it is given by Lederberg and Totem, and it is in this, what basically happens is that there are, if you see, these two bacterial cells are there. One is of where this F gene is present, so that is positive strain, and the other one is of negative strain, where this F gene or fertility gene factor is absent. So they how do they form this? So this is transferred by the formation of this bridge. So what basically is happening here is that this positive strain bacterium comes closer to this negative strain bacterium, and there is a bridge, the connection which is formed, and through this connection they transfer their genetic material, that is the non-nuclear genetic material, that is plasmid. So this plasmid, there are two ways by which it can transfer it into the other strain or the negative strain. So this and it can be either transferred directly, or it will make the copy of this plasmid, and then that copy will be transferred. So the original remains here, and the new copy which is formed here will be transferred to this negative strain. So in that case, what happens is that the positive will remain positive, and the other one will also become positive. Whereas the other possibility of this is that it transfers its own plasmid to the negative. So the positive strain becomes negative, and the negative strain becomes positive. Now overall, if you see that here, in the beginning there were two bacterium, and when they separate, then also it remains two. So that means there is no growth, no increase in number. So we say that it is a method of reproduction. Why? Because there is a recombination of genetic material, or there is an exchange of genetic material. But actually, if you see in the real sense, there is no reproduction, no increase in number of bacterium that is taking place, and that only happens when these two, they enter into the stage of binary fission. So this is how the mode of reproduction takes place in case of bacteria. It was asexually and sexually both explained. In the next part of the session, we are going to talk about the economic importance of bacteria, the benefits and the harmful effects of it. Okay, thank you, children.